All right, welcome to the Renal Regulation Challenge. This isn't your typical sit back and watch review. Nope, this is a rapid-fire test to see if you can really apply your knowledge when it counts. Let's see how you stack up. So, you think you've got renal fizz down cold, right? You've memorized all the parts of the nephron, you know the key hormones, but can you predict how they all dance together in real time? Let's find out. Okay, here's how we're going to play. The whole thing is super simple. I'm going to throw a factor at you. It could be a hormone, it could be a clinical state, and you've got just a couple of seconds to answer two key questions about it. So for every factor you see, you need to lock in two answers. First, does it lead to higher or lower reabsorption of a certain electrolyte? And second, where exactly in the nephron is this all going down? Simple enough, right? You ready? Let's do this. All right, first up, let's kick things off with round one. We're diving into the world of calcium, the king of cations. Get ready, your first factor is coming right up. Okay, first factor on the board, parathyroid hormone, or PTH. When those PTH levels shoot up, what happens to calcium reabsorption? Are we thinking higher, or are we thinking lower? And what's the location? Go on, lock in your answer. If you said higher, you got it. PTH has this really powerful hypocalciuric effect, which is just a fancy way of saying it forces the kidney to hang on to calcium for dear life. And where does it do this? Well, it's a double whammy. It boosts calcium reabsorption in both the thick ascending limb and the distal tubule. Okay, next up, we've got active vitamin D, PTH's crucial partner in crime. So what's its move? Does it crank calcium reabsorption up or dial it down? And what part of the nephron is its main stage? And the answer is higher. Again, its main playground is the distal tubule. You see, active vitamin D is like a construction foreman for gene expression. It tells the cells to build more of the key calcium transport proteins. We're talking TRV5 channels, calbindin, the PMCA pump. The whole crew works together to pull way more calcium out of the fluid and back into your blood. All right, let's switch gears from hormones to pharmacology. Imagine you've just given your patient a loop diuretic, something like furosemide. What's the immediate effect on calcium reabsorption? Is it going higher or lower? And where's the action happening? The correct answer is lower, and it's all going down in the thick ascending limb. Now, here's the why behind it. Normally, that part of the tubule has a positive electrical charge inside, and that charge literally pushes other positive ions, like calcium and magnesium, out into the blood. Well, loop diuretics come in and block a key transporter, and poof, that positive charge disappears. Without that electrical push, calcium and magnesium are just trapped in the tubule and get flushed out in the urine. And that's exactly why clinically we sometimes use loop diuretics to treat hypercalcemia. You're literally forcing the body to dump its extra calcium. Great job with calcium. You're doing awesome. Now let's move on to round two, the phosphate puzzle. And I'm calling it a puzzle because, as you're about to see, its regulation is tangled up with calcium, but in a way you might not expect. Okay, here we go. We saw what PTH does with calcium, but now what does PTH do to phosphate reabsorption? Higher or lower? And where? I'm telling you right now, be very careful. This is a classic board exam trap. And the answer is lower. This is just a beautiful illustration of PTH's two-sided mission. While it's busy saving calcium, it actively tells the kidney to dump phosphate. It's phosphaturic, meaning it makes you pee out phosphate. How? In the proximal tubule, it basically tells the cells to pull in and destroy the key sodium phosphate cotransporters. So the bottom line is, PTH raises your serum calcium, but it lowers your serum phosphate. That's an absolutely crucial distinction to remember. All right, next factor. Let's talk about a key phosphatonin. That's a hormone that regulates phosphate, called fibroblast growth factor 23, or FGF23. If FGF23 levels are high, what's that going to do to phosphate reabsorption? Higher or lower? And where's its site of action? The answer is lower. And just like PTH's effect on phosphate, this happens in the proximal tubule. Yep, FGF23 is another really powerful phosphaturic hormone. It teams up with its co-receptor, klotho, and sends a signal that does pretty much the same thing as PTH. It gets those NOPI code transporters pulled off the cell surface, which means, you guessed it, more phosphate gets excreted. Okay, you're on the home stretch. It's time for our final round, mastering magnesium. You know, magnesium is often the forgotten electrolyte, but its regulation is absolutely critical. Let's finish strong. Look who's back. Our old friend, PTH, one last time. We know what it does to calcium, we know what it does to phosphate, so what about magnesium? Does PTH make you reabsorb more or less? And the answer is higher. Turns out PTH wants to hold on to magnesium too, and its main spot for doing this is the thick ascending limb. Remember that paracellular pathway we talked about with the loop diuretics? That's where the bulk of magnesium reabsorption happens, and PTH helps make it even more efficient. Okay, this is it, the final question of the challenge. Let's think about a clinical state, hypermagnesemia. You've got way too much magnesium in the blood. How does the kidney itself respond to this? Does it try to reabsorb more magnesium or less magnesium to fix the problem? If you said lower, you are absolutely right. This is such a cool example of a feedback loop. When you have high magnesium levels in your blood, that high concentration actually suppresses its own reabsorption right there in the thick ascending limb. I mean, think about it. If the blood is already saturated with magnesium, there's just less of a driving force to pull more out of the tubule. The kidney just automatically lets the excess magnesium spill out into the urine to help get things back to normal. It's a really elegant system. And that's the challenge. You made it through. Fantastic work, seriously. Okay, now let's pull everything together and lock in this knowledge with a super high-yield summary. Okay, let's look at this all in one place. If you remember nothing else, remember this. PTH's opposing actions. It saves calcium, but it wastes phosphate. That's the board trap right there. Next, look at the thick ascending limb. What a critical hub, right? It's handling reabsorption for both calcium and magnesium, which is exactly why loop diuretics, which act there, cause you to lose both of them. And finally, notice the proximal tubule is the command center for phosphate regulation, with both PTH and FGF23 calling the shots there. Getting these relationships straight is absolutely key. So we've gone through all these regulators one by one, but here's the real question, the one that matters for clinical practice. What happens when these systems break? What clinical states could throw this entire beautiful, balanced system into total chaos? Think about things like chronic kidney disease, or a parathyroid tumor, or even genetic defects in these transporters. How would that mess everything up? That is your next challenge to think about. Thanks so much for joining me for this explainer.